Hello, my name is Michael Dabrowski, and I'm the CEO and also a co-founder of Pelago Bioscience. And today I have with me my colleague Stina Lundgren.
Yeah, my name is Stina Lundgren. I'm a principal project advisor at Pelago Bioscience. And today we're going to talk about how you can navigate your chemistry and explore your biology using CETSA. I'm going to start by telling you a little bit about Pelago. We're the home of CETSA. So this means that we're the specialized CRO focused on the CETSA technology. We have the exclusive control of the CETSA IP. We also have an in-house R&D team that are constantly developing the technology and applying it to new areas. So we focus on target engagement at Pelago. And this is the relevant measure of a compound binding to a protein target at the intended site. This is important to measure all through the drug discovery and development pipeline to make sure you develop safe and efficacious drugs. So that's why we have applied CETSA at all stages of drug discovery. Today we have done more than 240 projects. And that has been from early target identification to lead generation, but also working with projects in lead optimization and preclinical projects. We also supported some of the projects in clinical development. And the examples of application where CETSA can be used is quite broad. So that's from mode of action studies to screening up to generating relevant SAR for your compounds and lead optimization, to target validation, but also safety assessments. We see CETSA as one assay with two different readouts. It shares the same assay protocol, which is treat, heat, separate, and then detect. I'm going to talk about the CETSA Navigate, which is the targeted CETSA based on antibodies for the detection. And later, Michael will tell you more about the CETSA Explorer, which is the mass spec-based detection, which are profiling compounds protein-wide. CETSA works with basically any samples, and we have worked at Pelago with both human and animal, both intact and lysate cells, as well as solid tissue, xenograft, bacteria, plants, and even zebrafishes. So the principle of CETSA is that we look at the native folded protein and how this is melting upon applying a heat shock. So the principle is that if you have a compound, the melting behavior of the protein is affected. So you get a change in the melting temperature upon compound binding. And the readout I'm going to talk about first is based on the alpha technology, as you see here. So this is the CETSA high-throughput, which can be used for navigating your chemistry. It's a plate-based method. You're using dual antibodies for the proximity assay for detection. The compounds are unmodified, and we detect endogenously expressed protein in live cells, so there's no cloning required. And this altogether, of course, gives this assay a very high physiological relevance. So this is how the assay works. We first incubate the cells with the compounds. And this you can do in 96 well plates, but we usually work in 384 plate format. We apply a heat shock and lyse the cells. Then we transfer the lysate into detection plate and add detection consumables. And then we detect using the alpha technology. And by this, we can generate either melt and shift curves. So this is then profiling compounds at a single concentration, but then having a range of different temperatures. But we can also do the profiling at a single temperature and then do either concentration response curve or a single concentration screening. And this is an application of CETSA HG that has been used for primary screening. So earlier this year, Vertex and Astra presented on their primary screens using CETSA HG. Vertex used this on a quite challenging protein where there were no known binders to the protein when they started this project. They screened 50,000 compounds against it, and the CETSA HG screen could actually deliver some new chemical scaffolds for them. AstraZeneca has done a larger screen, so 500,000 compounds. They are basing their screen on the alpha LISA surefire detection method. And they have screened the compound versus CRAF. And they've been able to confirm both known CRAF compounds, but also compounds without known kinase activity. At Pelago, we have done a primary screen on CDK4 and STING. We validated this assay in THP1 and K562 cells. And from the validation, we could select a screening temperature based on the shift of reference compounds. We screened the library of 11,000 compounds. It was the same library against both targets. This was a focus library consisting of both lead-like compounds and fragments, but we also included a Prestwick library. We have done a pre-screening to test the robustness of the assay and to get to see what the robustness was, so the C prime calculations at different temperatures. And then after we embarked on the screening, which we did at 50 micromolar compound concentration in pre-spotted plates. This is the data from the pre-screen. And the CDK4, we selected to the THP1 cell as the cell matrix we were going to screening. We selected 46 degrees as the screening temperature, where we saw that we had the robust assay. For the STING target, K562 cells were selected, and the temperature of 51 degrees, where we also had a very robust assay. The compounds we profiled against STING were both agonists and antagonists. And quite interestingly, the agonists we screened had a quite large stabilization effect on STING, whereas the two inhibitors were both destabilizing STING. This is the screening result for CDK4, where we have a hit rate around 1%. Primarily, this is stabilizing compounds. And the next step, we will profile them most probably in duplicates, potentially also screening at the second temperature. For STING, we had not that many stabilizing compounds. The hit rate for the stabilizer was 0.2%, but we had quite many compounds that destabilize STING. So for the next step for this target, we're thinking about doing the hit confirmation by confirming these compounds at the second concentration, so a lower compound concentration. Another application of CETSA HD is to use it for hit confirmation. And here first we can see a correlation of the CETSA HD data with the biochemical FP assay. And this is highlighting how CETSA can be used following a biochemical or biophysical screen, where you would then confirm that your compounds are engaging your target in cell. And it can also then be used for filtering out compounds that have potentially low permeability or potentially not engaging the target in the cellular environment. The second graph showed the correlation between CETSA HD data and, in this case, a cellular PARylation assay. And of course, if you have a good correlation, this is something that strengthens your target validation. But also it highlights that CETSA HD can be used to identify silent binders. And this is particularly interesting if you are working on a PROTAC project where you would like to identify something that binds to a protein, but not necessarily affecting the function of the protein. CETSA HD can also be used for generation of SAR. So in this case, it's venirafenib and other BRAF inhibitors. And clearly the data can be used for them both prioritizing compounds, but also guiding the next compound design cycle. We have also looked in a study at Pelago on MEK1. And in this set, we have profiled a number of known MEK1 inhibitors, and that has both been compounds that bind to the ATP pocket, so they are ATP competitive, and also some allosteric compounds. And quite interestingly, the ATP compound destabilized MEK1, whereas the allosteric were all stabilizing MEK1. In the next step in this study, we looked if we could measure the inhibitor activity in parallel with the target engagement. So we looked at the RAS-RAF pathway, and initially we looked at the CETSA effect on MEK1, and this is then the concentration response with one of the allosteric compounds. Secondly, we looked at the phosphorylation level of ERK. So MEK is phosphorylating ERK. And if we inhibit MEK1, we would have lower amounts of phosphorylated ERK. And this is what we could see was done in a concentration response way as well. As a control, we also looked at the total level of ERK, which was not affected in this case. And we did the same also with the ATP competitive compound, where we could see that we had the same effect on the phosphorylation level of ERK, although this was destabilizing MEK1. We have also profiled a set of CEDK4 PROTACs using CETSA HD. And in this case, we have looked at PROTACs based on cereblon. They have the same linker, and one is then based on palbociclib, the compound A, and then the compound B is based on ribociclib. And both of these PROTACs were stabilizing CDK4 upon compound treatment, and this was also confirmed then in a dose-response manner. Lastly, we have also done the same profiling of this PROTACs against E3 ligase, so cereblon, and in this study we also included a third compound, which is also based on the same E3 ligase, so binding to cereblon, but this is then a BRD4 PROTAC. But all three PROTACs shown target engagement versus cereblon in this case, and then can be used for confirming that binding.
Thank you, Stina. And I'm then going to talk about the CETSA Explorer format. But before I dive into that, I'm once again going to say that the Navigate format that Stina talked about is one target at the time, but has the advantages of throughput. If you're interested, or if your favorite target wasn't on Stina's case studies, please visit our booth and we can talk about whether it's in our catalog or we should develop a new assay for you. But what you can't do then is to look at more than one target at the time, and that's the CETSA Explorer format, because that's a proteome-wide profiling of few compounds against the whole proteome. The setup, the assay method is still treat, heat, separate, detect, but the detection mode after the heat shock and centrifugation is now using an LC-MS mass spectrometer. And what we get there is still the same format of melt curves with and without the compound. And if the compound induces a thermal stabilization or a thermal destabilization, that means that the compound is engaging with that protein of which we're looking at the melt curve. And the output is then one of these volcano plots that you see on the right-hand side of the slide, showing stabilization or destabilizations, and then on the y-axis, the significance of the hit. The kind of data you get when you do these studies is hopefully a whole lot of nothing, meaning that if you profile your compound, you ideally don't want it to engage with all the proteins in the cells. So, and that's also what we see, that the majority of the proteins are unaffected by the ligands that we test. But then you see some direct binding events. You see them hopefully on your intended target, but also on unintended target or other targets, whether they are liabilities or not, that's of course up to you to discriminate between them. But primary binding events. And then because the cell is alive and intact, you will also have downstream effects such as phosphorylations that Stina just talked about. dimerizations, translocations, etc. All these changes in the functional status of proteins in general also leaves a mark on the thermal stability of that protein and is therefore picked up in this assay. But because it picks up both, you need something to discern between a direct binding event and a downstream event. And the way to do that is that we typically do two experiments. One in a pre-lysed sample, so you add your compound after you lyse the cells. And in that situation, in general, biology will be off, and you will see fewer number of hits and shifts. With the cell being alive, the volcanoes become more active because the biology is on, and there you see both the pathway hits and the direct binding events. And ideally, subtracting those two data sets, the difference being then your signaling pathway or your mode of action of the compound. What kind of processes does that compound initiate in the cell? As a real life example, we took staurosporin and profiled in intact cells. And you can see that this volcano plot is quite active. Staurosporin is a promiscuous binder. And it only gets worse or more interesting when you then look at the intact cell, because there is, of course, proteins downstream, which are affected by the primary binding of staurosporin. Putting it up in the way that we're usually or customized to look at kinase selectivity trees is one of these family trees. And you can see here that is then our data set in a single run. We've been able, in the same method format, to show the known selectivity targets of staurosporin, confirming what we already know, but in a single run. And the correlation is actually quite good, which is slightly surprising for me because this is a biophysical assay, but you can compare the XC50 values from the CETSA experiment with KD values from normal or established competitive binding assays. And as you can see, there's a fairly good one-to-one correlation, but of course with a drop-off when you look at the CETSA values. More on the mode of action, the pathway. We looked at the IMiDs, thalidomide, pomalidomide, lenalidomide, etc. Originally developed as an antiemetic compound back in the 50s and having these detrimental birth effects. But it is also used in therapy nowadays, and one of the applications are in oncology. The mode of action, which is cartooned on the lower part of this slide, is the result of many clever scientists doing many kinds of studies to discern, well, what is the mode of action of the IMiDs, and that is now well established that the IMiD binds to cereblon. The ligase activity is activated, ubiquitinating then the substrates, and then they are degraded by the proteasome. We then did the experiment that I showed you now twice. So this is the third time of profiling a compound, both in the intact cell and in lysate. And so on the y-axis here, we now have the lysate amplitude, and on the x-axis, we have the intact cell amplitude. And you can see cereblon up here on the right-hand corner. The interpretation is then that the compound binds to cereblon, and that is something that we see both in lysate, so it is a direct binding event, and we also see it in the intact cell. In contrast, the Aiolos-Icaros proteins here don't move very far on the lysate axis. It's a logarithmic scale. They move a lot further on the intact cell scale. So the interpretation is that this is not something that happens to a significant extent in lysate. It only happens to a significant extent in the intact cell. So therefore it is a downstream effect. and exactly like we saw in the cartoon. So in popular terms, you can say that in 2 experiments, we reproduced about 20 years of research, but that might be stretching it. But at least we see this mode of action in these 2 experiments. So it was a whistle-stop tour of case studies, but what Stina told you was that CETSA HT can be used for primary screening, but also generation of tools if your target doesn't have good tools. Hit confirmation, SAR, and indeed translational studies. And because of the sensitivity, throughput, and robustness, then you can identify both very low affinity binders, but of course also high affinity binders. And she then lastly showed you about using CETSA HT for profiling both the target protein of a PROTAC and the ligase. And what I rattled through was that CETSA MS is good for proteome-wide profiling of both the direct binding events and the downstream cellular pathway effects that you see in intact cells. And therefore, the applications of CETSA MS will include both selectivity and specificity studies. You can validate your tool and probes, and of course, benchmark your candidates against other candidates or other compounds out there, and start deconvoluting the mode of action of your compounds. If we didn't say it in this talk, well, you can visit our booth here at this meeting and get to these targets that we've worked on, or you can book a meeting with us to discuss if those targets weren't in the catalog. And then tomorrow, on October the 7th, there will be a panel discussion which I will moderate, and there we hopefully will discuss more on good lead generation practices, and well, we'll see if the panel also discusses CETSA. So back to us and the rest of the team. Pelago, we're the home of CETSA, and we are that happy on the picture almost all the time.
Yeah, that's true. Thank you.
Thank you.